And the topic will be regulatory T cells. By the way, can you see my cursor? Or?
Yes, we clearly.
Oh, okay, great. Okay, so during the talk, I would like to explain the role of regulatory T cells in infectious diseases, and this research is actually quite young research area, because and I will go through the history. The role of regulatory T cells in infectious diseases was far from being clear, and it was discussed a lot when we started to work on this. So what's the basic idea? Why do we think that we need something like suppressing T cells? So the immune system is tightly regulated because, as you know, if you have an overshooting immune activity, you develop autoimmune diseases. So it's very important to control every step of the immune system. Otherwise, it can be really harmful for the own organism. So there was a basic idea already in the 80s that cytotoxic T cells, effector T cells, have to be controlled by another cell type, that they don't have overshooting reactions and that they are not harmful. So in the 80s, people called that suppressor T cells and developed the concept of suppressor T cells. So Richard Gershon already postulated that actually in the 70s, and then in the 80s there were a number of experiments about this. And if you look at textbooks from the 80s and then early 90s, there's actually always a chapter about suppressor T cells that are claimed to be CD8 positive T cells. And so these T cells were studied a lot. However, there was no marker for these T cells. So nobody could identify those T cell populations clearly. And so after a couple of years of research, a lot of immunologists started not to believe anymore in suppressor T cells. And like I said, the main problem was that no molecular marker was defined for these cells. So then from like 1985 to 2000, hardly any immunologist believed in suppressor T cells anymore. And if you would say you work on these cells, you know, people would really ban you from conferences and not talk to you anymore, because there was a strong disbelief in these cells. And one person who was still working on those was Shimon Sakaguchi in Japan. And he published 1995 a groundbreaking paper that changed the field again completely. And he showed that there's a special population in his work, not CD8 positive T cells, but CD4 positive T cells that can suppress effector T cell responses. And he also defined a marker for these cells, and that was the IL-2 receptor alpha chain called CD25. So he said that all these suppressing CD4 cells are highly expressing CD25, and that this is their marker. But he was so afraid that people would dislike his research results that he didn't call them suppressor T cells, but he called them regulatory T cells to find a different name. And his first paper in 1995 opened a whole new area of research. And then a lot of people jumped on this topic, and now we have thousands of papers on regulatory T cells. So what did Shimon Sakaguchi do? And let's start down here with these experiments. Those are cell culture in vitro experiments. So he isolated effector CD8 cells or effector CD4 cells and stimulated them in vitro so that they proliferate and produce cytokines. And he did that without adding CD4 cells that are CD25 positive, or he isolated these cells and added them to the culture. And what you saw was when he added these CD4 positive, CD25 expressing cells, which he called Tregs, then he could inhibit the proliferation and cytokine production of these effector cells, indicating that there's a special population of cells that can suppress effector T cells. And he did a very similar experiment in the mouse. He took nude mice that don't have any fur, and they not only have any fur, they don't have any T cells as well. And then he added T cells from wild-type mice into these animals, and they behaved normal. They did not develop any disease. However, if he did the same experiment, take these T cells out of wild-type mice, and then he separated out the red ones here, the CD4, CD25 positive ones, and transferred only the effector T cells, then these animals developed autoimmune diseases, indicating that also in vivo, these red cells, these CD4, CD25 positives are very important to control the effector response of these T cells. Okay, so these were the key experiments to show that there is a population of suppressing T cells. And as I said, he called them regulatory T cells, or in short, Tregs. So now in the current literature, we define two different populations of Tregs. And the first one is called natural Tregs, or now more and more called thymus-derived Tregs, because that's where they come from. So they are directly educated in the thymus and come directly from the thymus as CD4, CD25 high. So they express a lot of the IL-2 receptor. And they make up about 5% of the CD4 T cell pool. So 95% are effector CD4 cells, 5% are the counter-regulators, the regulatory T cells. And they can suppress CD8 positive cytotoxic T cells or CD4 positive T helper cells. And they can inhibit their activation, their proliferation, and their cytokine production and their cytotoxicity. So they can either regulate and suppress their activation and expansion, and also their function. So what are the mechanisms that have been described? So when these cells were discovered, the million-dollar question was, what is the mechanism of their suppression? And everyone was working on this. But it turned out they don't have a single mechanism. They have a number of different mechanisms how they can suppress effector T cells. And this just lists the most important ones. So they can, one mechanism is that they produce cytokines that are inhibiting other immune cells, like TGF-beta. That's an immunosuppressive cytokine. On the other hand, they have cell contact dependent mechanisms that they can work with. And one is CTLA4, a well-known inhibiting ligand for T cell proliferation. So they can express CTLA4 and then interact with CD8 cells. They then are impaired in their proliferation. And the same is true for inhibitory ligand, which is called PDL1, that interacts with PD1 and shuts down CD8 responses. They can also consume all IL-2. I told you they have a lot of CD25, so the high affinity IL-2 receptor. And if IL-2 is in the area, they consume all IL-2. But effector T cells also need IL-2 for their development. So when they take all the IL-2 away, the Tregs, there's nothing left for the effector cells, and they can't really produce their function then. And there's one other mechanism. Those are enzymes that they express on their surface, CD39 and CD73. These two enzymes convert ADP to AMP. And cyclic AMP is a suppressor of T cell responses. So with their enzymes on the surface, they can convert molecules to T cell suppressing molecules. And all these different functions have been described in different suppressing models, in cancer models and infectious disease models and so on. So there's no single mechanism, but it's a number of different mechanisms. So what are their characteristics in terms of markers? And I told you they express a lot of CD25. That was their initial way to be characterized. And then they also express a number of other molecules, but not exclusively. So all these other molecules that you see here are also found on some effector cell populations. And they might be highly expressed on Tregs, but it's not a unique marker for Tregs. And then also Simon Sakaguchi discovered a unique marker for Tregs, and this is FOXP3. FOXP3 is a transcription factor, and this is absolutely essential for starting the regulatory program in T cells. So without activating this transcription factor, T cells are not becoming regulatory T cells, and they are not suppressing. So at least in mice, this is really a unique marker. If a cell is FOXP3 positive, you know it's a regulatory T cell. Unfortunately, in humans, it's not that black and white. It's also a very good marker in human cells, but there are some very few effector T cells that also express FOXP3 in humans. Okay, so these natural Tregs can expand in vivo in the mouse. This was a big puzzle in the beginning of the research because it's impossible to expand regulatory T cells in vitro. So in the cell culture system, you cannot expand them, or it's very difficult. Now there are a few protocols, but when the research started, it was impossible. However, in the living mouse, this was shown in this model where the scientists used T cell receptor transgenic CD4 cells that were CD25 positive, and they were specific for an OVA antigen. When they transferred these regulatory T cells in a mouse that expressed OVA in all body cells, so a transgenic mouse, then these OVA-specific Tregs massively expanded and started to shut down all effector T cells in this mouse. So this was the first experiment to show that if they see their antigen in vivo, they can expand. And just like other T cells also expand when they see their antigen. And that was a lot different, as I said, than the first in vitro results, where people concluded from the results that Tregs cannot expand, cannot proliferate. But these in vivo experiments showed that this is not true. So the second population of Tregs are the so-called induced Tregs, or now they are mostly called peripheral induced Tregs. So here we have a different situation. There's an effector cell that came from the thymus, and it was born as effector cell. But then some circumstances convert this effector cell to a Treg. And this usually goes along with the expression of FOXP3. So the FOXP3 program has to be turned on. And then, since they don't come directly from the thymus, we call them induced Tregs or peripherally induced Tregs. And they came, like I said, from effector T cells. So it's a different origin for this cell population. So what's their mechanism of suppression? And there are a few other mechanisms that have been described for these cells. The most important one is the production of IL-10. IL-10 is an immunosuppressive cytokine, and these induced Tregs have a strong ability to produce IL-10. They also produce TGF-beta and can suppress by TGF-beta. And then there are a number of papers, mainly from one group in France, showing that they can produce cytotoxic molecules, especially granzyme B, and not only suppress effector T cells, but really kill them. But this has not really been found in other labs, including ours. We could never verify this data. So I'm not sure if this mechanism of action is really important or true. So what are their characteristics? And here it's even more complicated than in the natural Tregs because they have a number of markers, but they are also shared with some effector cells. Most of them are FOXP3 positive, but not all of them. There are some FOXP3 negative induced Tregs. And they are not all positive for CD25. So that's not an obvious marker for this population. So they are hard to define. They are mainly have to be defined by their function and cannot be defined by molecular markers. Okay, so this is the current concept of these two populations. So here are the natural or thymus-derived Tregs on the right. And we think that in most systems where they were studied, they are actually specific for self-antigens. So they see constantly self-antigens and are driven by the T-cell recognition of self-antigens that are always present in the organism. Whereas induced or peripherally induced Tregs, they recognize foreign antigens, for example, viral antigens or bacterial antigens or neoantigens of tumors, for example. So tumor antigens can be self-antigens or they can be very rare neoantigens that are expressed because of mutations in tumor genes. So that's the current concept, that they have different sources of antigen that they recognize. And I will come back to that in a minute when I explain what we are working on. So that's also the current concept in virus infections. So in virus infections, you have virus production, and then effector T cells, cytotoxic CD8 cells or CD4 cells, recognize viral antigens and have this clonal expansion of T cells that is characteristic for T and B cells when they recognize their specific antigen. And that leads to antiviral activity and massive reduction of the virus, or even elimination of the virus in the best case. However, when the virus load goes down, you still have these massively activated CD8 cells, and that's the point when they have to be controlled, or they can actually make more damage than the virus itself. And this is when the regulatory T cells have to come in place and then start to suppress the further proliferation or the function of the effector CD8 cells or CD4 cells. And these Tregs might also be driven by the virus or by other mechanisms, and I will come back to that in a minute. So, since this research started in 2000, the research on regulatory T cells in infectious diseases, for almost all infectious diseases, there was a Treg response after infection was demonstrated. And this list is not complete. Actually, the first viruses that this concept was shown for was Friend virus, and I will come back to that in a minute. That's a mouse retrovirus, and human herpes virus, herpes simplex virus. So these both papers appeared at the same time. And then for parasites, Leishmania Meyer was the first model where this was shown that Tregs play an important role in infectious diseases. And this is how we think this knowledge, and if we know more about Tregs, can be used therapeutically. And there are completely different strategies for different diseases. So here we have diseases like inflammation or graft versus host disease in transplant patients, for example, or autoimmune diseases. In that situation, you have too many effector cells and too few regulatory T cells, so that you have an imbalance between the effector T cells and the Tregs, and the effector T cell, the Tregs are not frequent enough to really suppress effector responses. So in that scenario, we would like to expand the population of regulatory T cells and have more of those so that they are able to suppress effector T cells to prevent then autoimmunity or graft versus host disease. So this would be the resolution then. We have equal numbers of Tregs and effector T cells. So in chronic infection and cancer, the situation is completely different. Here we have too few effector cells and we have too many regulatory T cells. And the regulatory T cells suppress the effector cell responses, with the consequence that the tumor can't be rejected or that the chronic infection can't be cleared. So in that scenario, we would like to do something different. We would like to reduce the numbers of Tregs in order to enable the immune system to have more effector cells and then fight the cancer or fight the chronic infection. And at the end of the day, it would also be nice then to have an equal number, probably, of Tregs and effector cells. And then we also have a problem in allergy. There also we have a special type of effector cells, the Th2 effector cells, that are too many and induce allergies, overshooting immune reactions. And we have not enough Tregs to suppress that. And in that scenario, we would also like to have more Tregs to suppress the Th2 responses that lead to allergy. So you can see for different diseases and problems, Tregs play an important role, and we would like to follow different strategies to either increase their numbers or reduce their numbers. And there are a lot of clinical trials also trying to achieve that. And I'm pretty sure once we have more of that in the clinic and can really use that for patients, then Shimon Sakaguchi will receive the Nobel Prize for his groundbreaking work in 1995. Okay, I will now jump in and talk about our own research, which we started around the year 2000. Like I said, Tregs were discovered in autoimmune diseases, and at the time, 1995, it was completely unclear if they play any role in infectious diseases. Actually, most people said this is highly unlikely. Because what you want in an infectious disease is you want a strong effector T-cell response, and you don't want anything that counter-regulates this. But in fact, that's not true. You need counter-regulation so that the immune system does not harm the organism itself. So, like I said before, we have a mouse model here in the lab, which is called Friend virus. And the name comes from Charlotte Friend, a Swiss virologist, and she discovered this virus in mice. And the mice, this virus can induce leukemia in susceptible animals. However, if you take certain mouse strains, for example, B6 mice, then they don't develop leukemia because they make a very strong immune response against the virus and can control acute infection. However, they cannot eliminate the virus and become chronically infected for life. So it's a very nice model to study chronic virus infection in mice, and also to study chronic retroviral infection because it's the same virus family that HIV belongs to. So we use this model in order to ask the question, what is the role of regulatory T cells in the development of chronic lifelong virus infections? And you probably know that a number of lifelong virus infections are causing medical problems. For example, HIV, but also hepatitis B and hepatitis C, and also some herpes virus infections that can reactivate later in life. Okay, so I told you the role of Tregs in virus infections was first published in the year 2000 in the herpes simplex model and in the Friend virus model. This is the Friend virus paper from my friend Kim Hasenkrug from the US. I worked for quite some time in his lab, and he was the first one to discover this. And what he showed was that Tregs massively expand in a chronic virus infection. And we then, after a number of years and more research, understood that these Tregs prevent immunopathology caused by overshooting immune responses. And this was shown for different virus infections now, including HIV and herpes viruses and also influenza. So when we started this research, we took advantage of a very nice mouse that was established by a German group, Tim Sparwasser. And this was especially a very nice tool to study regulatory T cells. So what Tim did, he generated a transgenic mouse in which all regulatory T cells and only exclusively the regulatory T cells express a fluorescent marker, GFP. So all Tregs are green in these mice, and only the Tregs are green. And these Tregs also express a receptor, which is the diphtheria toxin receptor. Diphtheria toxin is really dangerous for humans because we all have the diphtheria toxin receptor on our cells, but mice don't have the diphtheria toxin receptor on their cells. It's not expressed, so mice are resistant against diphtheria toxin. However, so this transgenic mouse expresses under the control of the FOXP3 promoter, GFP, to make all cells green, and diphtheria toxin receptor. So if you inject diphtheria toxin into these animals, all the Tregs are selectively killed because only they express this receptor. So you can see that here. First of all, here you can see that all these FOXP3 positive cells, they express GFP, so they are green. And then if you look at the Tregs from this mouse, if you don't do any treatment, you can see here the Tregs that are green and are 13% of all CD4 cells after Friend virus infection, for example. So if you inject diphtheria toxin into these mice, then you can see that all Tregs are selectively killed. There's 0% left. So a very nice tool to really study the role of Tregs during an infectious disease in the mouse. And this is what we took advantage of, and you will see data from this model. So the model is called DREG mouse. So that stands for depletion of regulatory T cells. Okay, so this just shows you, illustrates the expansion of regulatory T cells after Friend virus infection of the mouse. So when you infect the mice, this is the population of Tregs. So in terms of percentage of Tregs from the CD4 compartment, and in terms of absolute numbers of Tregs, this number goes up and it stays higher than at the starting point. And this is true for liver. This is true for the spleen. But not really for other organs where you don't really have virus replication. So the expansion of Tregs you only see in organs where the virus is replicating and where you have high viral loads. Okay, so one of the first questions that we tried to address was what was driving this expansion of regulatory T cells. So if a virus infection starts and you see then regulatory T cells expanding, what is driving this expansion? And the first idea obviously was the virus itself, the viral antigens. And we studied this for a long time, and I'm not going to show you all the results, but the results were no, it's not the virus, it's not viral antigens, because the main population of expanding Tregs were natural Tregs, and I told you they are self-antigen specific. So they don't see viral antigens, and actually the virus antigens didn't play any role in the expansion of regulatory T cells. So this one slide summarizes a lot of data. And I start down here because this is easy to understand. So when you have a virus infection, you have an expansion of effector CD4 cells because they are virus specific. They recognize the viral antigens and they have a clonal expansion and have an effector CD4 T cell response. These effector CD4 T cells produce a number of cytokines, and one important cytokine that they produce is IL-2. And I told you the regulatory T cells express a lot of the IL-2 receptor. That's their characteristic, CD25 high. So if the effector CD4 cells express IL-2, then this drives the expansion of regulatory T cells because they are CD25 positive. So this is pretty easy. They are self-antigen specific, so their first antigen contact is just constantly seeing self-antigens, and if they then receive a lot of IL-2, the Tregs expand. So the effector cells themselves, by producing IL-2, drive the expansion of their counter-regulating cells, the Tregs. So that was one part of it. But then we realized there's a special population of Tregs, at least in mice, and they all have the same beta chain of the T cell receptor, the beta chain 5. And they are a large population of expanding Tregs after a virus infection. And we didn't know what was going on, but we found out they were not dependent on IL-2. They did not respond to IL-2, interestingly. And then after a long work, we found out how they are expanding. And this was very interesting because what they were specific for was a self-antigen, which was a very special self-antigen. It was an endogenous retroviral antigen. You probably know that we are full of endogenous retroviruses. 6 to 8% of our genome, of our whole genome, is endogenous retroviruses. And we still don't know what a lot of these antigens do, but here's some example what they do, at least in the mouse. So all mouse strains express this endogenous retroviral antigen, which is called mouse memory tumor virus 9 superantigen. So this is an ancestor from a virus infection, and it's still in the genome of all mice, and this antigen is being expressed. And a large population of the Tregs is specific for this superantigen. And this is their T cell receptor contact to the superantigen. And then they don't need IL-2, but they need a different cytokine, and that doesn't come from CD4 cells, but it comes from CD8 cells. And this is membrane-bound TNF-alpha. So when CD8 cells become activated after a viral infection, they make TNF-alpha. Some of the TNF-alpha is membrane-bound, and that interacts with the TNF-alpha receptor on regulatory T cells and on the special population of regulatory T cells. And then they become larger and larger and are expanded by this cytokine. So interesting enough, effector CD4 cells can expand regulatory T cells via IL-2, but also activated effector CD8 cells can expand a certain population of Tregs, and for that they use TNF-alpha, not IL-2. But both populations of Tregs are self-specific. Here we know the antigen. It's a superantigen from an endogenous retrovirus. Here we don't know exactly what self-antigens are involved. But this makes a lot of sense in terms of biology, because that means you have the effector cell responses first after a virus infection. So they can do the job first, can be cytotoxic, can produce effector cytokines here. And then if they are expanded, they start to expand regulatory T cells, which then counter-regulate their response. So in terms of biology, in an infectious disease and the time course, this makes a lot of sense. Okay, so what are these cells suppressing? Once they are expanded, what's their suppression? And in the beginning, we looked at CD8 responses during an acute infection. So the question was, during an acute virus infection, do the Tregs that expand, do they suppress CD8 responses, cytotoxic CD8 responses? And the answer was yes. We showed that in the DREG mouse model for the first time, that they suppress the proliferation and effector function of cytotoxic CD8 cells. And this is only summarized here in one slide. So the second question was, do they also suppress virus-specific CD4 cells, not only CD8 cells? And so we did the following experiment. We took the DREG mice where we can deplete regulatory T cells, infected them with friend virus, and depleted regulatory T cells, or let them undepleted, and looked for the CD4 response in these animals. And this is shown here. So this is CD4 cytotoxicity. Can they produce cytotoxic molecules after virus infection? And you can see here, these are uninfected mice. If you infect them with friend virus, they produce a little bit of cytotoxic molecules. We see that in almost every infection. Cytotoxic CD4 cells are very much controlled by the immune system because they can be very dangerous. Usually only CD8 cells are cytotoxic. CD4 cells should not be cytotoxic. So they are controlled, and part of their control is by regulatory T cells. So if you deplete regulatory T cells, then you have more granzyme B producing CD4 cells. However, really a striking difference you only find if you in addition deplete CD8 cells, telling us that CD4 cytotoxicity is under the control of Tregs and effector CD8 cells. Only if you take both away, then you have this really strong cytotoxic response, and we can also measure that in a real killing assay. So not only look for cytotoxic molecules, but really look for killing in the mouse. And I will show you in a minute this so-called in vivo CTL assay. We can do that for CD4 cells and for CD8 cells. And I show you how it works in a minute, so you can really measure cytotoxicity in the mouse. Okay, and when we look for viral loads, does that have any effect on viral loads? And I walk you through here. So you can see these are the viral loads in a just friend virus infected mouse. If you deplete Tregs in this animal, the viral load goes down. And the reason is that you have more activated CD8 cells in these animals. And we showed that in the previous paper. If you deplete CD8 cells, then the virus load goes up, showing you that CD8 cells are really important. So if you deplete Tregs and CD8 cells, then the virus can be controlled much better because now you have cytotoxic CD4s and they control the virus. And if you deplete also these CD4 cells, in addition, deplete everything, then the virus load goes back up, showing you that indeed you have cytotoxic CD4s that do this virus control here. And they are under the control of Tregs and CD8 cells. Okay, this was acute infection. One important question was, what about the chronic infection? And there's an important hallmark of chronic infections that you find almost in every chronic infection, and that's the so-called T-cell exhaustion. That means that in these infections, and you can take HIV or hepatitis or some herpes virus infections, when you infect animals or also humans, you find that also in humans, you see an antiviral effector cell response. But then if the virus is not cleared and in these infections the virus is not eliminated, then the T cells start to develop a dysfunctional phenotype. They are still there. You can still see them and measure them, but they can't do anything anymore. And this is has been shown nicely in this review. So this is a functional T cell at the beginning of the infection that can make interferon gamma, TNF-alpha, IL-2, and has a cytotoxic potential, so can kill virus-infected cells and can proliferate. But then, if this chronic infection goes on, they start to partially lose their functions. And the first thing that's lost is the cytotoxic activity. Then they lose the ability to produce IL-2. Then they also lose the TNF-alpha production. And at the end, they also lose the interferon gamma production. So then you have these ghost cells that are still there, but they cannot proliferate anymore and don't have any of their initial functions. And they eventually can be depleted. But I think this last here is a little misleading. That doesn't happen in most infections. So in most infections, you have this state of full exhaustion during the established chronic infection. So the question was, is this somehow influenced by regulatory T cells so that these T cells develop functional exhaustion? So we addressed that in the Friend virus model as well, and this just shows you a picture that we also have functional exhaustion of effector CD8 cells, for example, in the Friend virus model in chronic infection. You can see here the granzyme production of CD8 cells, activated CD8 cells, and you can see here they produce granzyme, at least 10%, then at 15 days early on in infection, at 15 days post-infection, this is already a lot less. And if you look at chronic infection, you still find these CD8 cells. They are still activated, but they can no longer produce granzyme B. It's only a very small fraction. So we have functional exhaustion in chronic Friend virus infection. So the question was, what is the role of regulatory T cells in this phenomenon? So we again used the DREC mice where we can deplete regulatory T cells. We infected them with Friend virus and then just let them sit 60 days to develop chronic infection. And then after these 60 days, we started to deplete regulatory T cells and compared them to mice that were not depleted. And this is the data when you just look at the numbers of cells. So here's always the chronically infected mouse. These are activated CD8 cells. And you have a lot more of these activated CD8 cells if you deplete regulatory T cells. And these are different organs. And tetramer positive cells are virus-specific CD8 cells. So they recognize the virus and are specific for the virus. And if you deplete regulatory T cells, you have more virus-specific CD8 cells. So Tregs can suppress the proliferation and activation of CD8 cells, but the most important question was, can they suppress the function of these CD8 cells? Because we were interested in the CD8 T cell exhaustion. And the picture was black and white here. So this is chronically infected mice, all these down here. So you can immediately see in chronically infected mice, the CD8 cells cannot produce IL-2, TNF-alpha, or interferon gamma, just like I showed you from this review before. If we then deplete regulatory T cells in these animals, they start producing IL-2 again. They start to produce TNF-alpha again, and they start to produce interferon gamma again. The second question was, do they also start to produce cytotoxic molecules when we deplete regulatory T cells, these CD8 cells? And this shows you that they are not able to really produce granzyme B. in chronic infection. However, if you deplete Tregs, they are reactivated in their production of granzyme B. And then we used this assay where we can study cytotoxicity and really looked for cytotoxicity in these mice. And this is the situation of chronic infection, and that these both figures are similar, tells you that there was no cytotoxic activity, no killing in chronically infected mice. However, if we depleted regulatory T cells, then these mice were able to kill target cells. And I will show you in the next slide how this assay works. And this is the percentage of killing in different organs, in the blood, in the lymph nodes, in the spleen. So we could show that with depleting regulatory T cells in these chronically infected mice, we could reactivate the exhausted CD8 cells. That's one big issue for cancer and therapy of chronic infectious diseases. The ability to try to reactivate the cytotoxic CD8 response, because also in cancer, this is dysfunctional. Okay, so this is the assay. To brief you, explain to you what an in vivo cytotoxicity assay is. So for a cytotoxicity assay, you need targets that can be killed by the CTLs. And as targets, we just take lymphocytes from the spleen or lymph nodes of an uninfected naive mouse. So these are the potential targets. And then to make them targets, they need to be loaded with a peptide that can be recognized by the T cells. So what you need to know is a T cell epitope, the immunodominant T cell epitope in this infection. And then you just order the peptide, the epitope peptide, and you just incubate the cells with this peptide. And the peptide is loaded on MHC class 1 molecules automatically. You don't have to do anything for that. You just incubate them together. And then you need a control, which is either loaded with no peptide, CTL epitope peptide, or you take an irrelevant peptide that can't be recognized by specific CD8 cells. But you need a 1-to-1 ratio of these two cells, and then you label them with a fluorescent dye. So here in this case, this population is labeled in red, and this population is labeled in blue. Then, as I said, you mix them 1-to-1. One is a real target, and one is the control. And then you transfer them into your animals of interest, chronically infected mice or chronically infected mice in which the Tregs were depleted, or naive animals as control. And what you can then follow is the killing of this population of cells. So you have to transfer them in, and then you have to kill the mice after a couple of hours, and then analyze the number of cells that you find in these animals. And that gives you, in the comparison between these two populations, a direct number of killed cells. So this is the situation. If no cells are killed in both groups, then you have the same number of cells that you re-isolate from the mice. If you have killed cells, then you have a lower number of cells here in the peptide labeled group. That means all the other cells were killed by CTLs in this time period. So that's an in vivo CTL assay that we do a lot for these experiments. So the last question in these chronically infected mice was, if we deplete regulatory T cells and reactivate the CD8 cells, does that change anything with the viral loads in these animals? So do we have less virus? And the answer is yes. So these are, again, two different organs. This is the normal, already very low viral load in this chronic friend virus infection. However, if we deplete regulatory T cells, then the viral load goes down by 10 to 15 times. So we were able to reduce, the viral loads in these animals with this Treg depletion, showing that Treg directed therapy would be a possible therapeutic approach to treat chronic virus infections. However, what you also see here is that we still find virus in all the animals. It's very low, and it's in some animals, it's only one infected cell in 1 million uninfected cells. But still, if you have a very sensitive assay, you can still find virus. So this therapy does not lead to elimination of the virus, which we would like to achieve in chronic infections. Jump over this. So that's why we thought, okay, maybe we combine this therapy with another therapy, which was developed by Rafi Ahmed in the US, who was also working on these exhausted effector T cells. And you probably know the story because it was very famous, and it's now being used in cancer therapy in the clinic. And this is the story of blocking inhibitory receptors. So effector T cells have a number of receptors. When you trigger those receptors, you can downregulate the function of T cells, of effector T cells. And the most prominent one is PD-1, because that's being targeted in cancer therapy all over the world now. So if you block the receptor ligand interaction with antibodies of PD-1, and the ligand is PD-L1, then you can actually reactivate some of the function of T cells and make them anti-tumor reactive in cancer patients. So obviously, this exhaustion of CD8 cells that we see in cancer and chronic infection is driven by two different mechanisms. And this is the inhibitory receptors that I show here, and by regulatory T cells. So our idea was, let's combine these two therapies and see if we can really achieve elimination of chronic virus. And this is shown here. Actually, we not only blocked PD-L1, but also, TIM3, here another inhibitory receptor that you see down here. So it was already a combination therapy of different antibodies. And at the same time, we depleted regulatory T cells. So again, we used the Treg mouse, initiated chronic infection, waited 60 days after infection, and then we started this treatment to target the Tregs and the inhibitory receptors at the same time. And here's the data in terms of CD8 T cell responses. So here's the number of activated CD8 cells that we have in a chronically infected mouse. If you deplete Tregs, this goes up. If you inhibit block the inhibitory receptors, this goes also up. If you combine these two therapies, you have even a further significantly enhanced response here in terms of CD8 activation. And these are the numbers of virus-specific CD8 cells. And again, this is increased by Treg depletion. I showed you that before. And this is also increased if you block the inhibitory receptors. However, there's an additional effect if you combine these two therapies. And then it was obviously very interesting what happens to the viral loads in these animals. And here are the normal viral loads of a friend virus infected mouse. If you deplete regulatory T cells, then this goes down. If you block the inhibitory receptors, this has a higher effect. So this pathway seems to be more important. However, if you combine these two therapies, you have the strongest effect. And for the first time, we had at least 3 mice out of these, I don't know, 12 or so that we did, where we could not find any virus anymore. So for the first time, we had the first animals where we eliminated the chronic virus with this combination therapy. So that's obviously something that we would like to further improve, because 3 out of 12 is still not perfect, but we never saw that before, that we were able to eliminate chronic friend virus and retroviral infection. Okay, so the last part of the talk, only a couple of slides. I show you something that maybe Matthias Gunzer, doing his last talk in this academy, will come back to, and this is imaging. So what we are trying to do now is we want to see the interaction and the movement and the function of these cells in the living mouse. So until now, all the experiments that I showed you was we infected mice, manipulated their immune system, then we have to kill them and do flow cytometry to look at their immune cells or to look at their viral loads. But obviously we would like to see the action of immune cells in the living mouse and learn what is going on in a living organism. So in order to do that, Matthias Gunzer has established so-called intravital 2-photon microscopy. And he established that in the bone marrow. And he was the first worldwide to do that. So you have the living mouse in this water bath. And you can image the interaction of immune cells in the bone marrow. You have to take away the fur and take away some of the muscle to go down to the bone here, the fibia or the tibia. And then you have to thin the bone here, so that it's not open, the bone, but thin enough that you can use the 2-photon microscopy objective to go through the bone and film the cells in the bone marrow. And this is what Matthias established, and we together with Matthias for the friend virus model. And you can see cells here already. So these are red fluorescent cells. You can see here the bone, that's the thin bone. And you can see that we can do microscopy inside of the bone. And these red cells here are, for example, fluorescently labeled CD8 positive cytotoxic cells. So we can see them in the bone. So, by the way, why did we use bone marrow? Bone marrow is a very important organ of friend virus replication. So if you inject the virus into mice, a lot of the virus goes into the bone marrow and replicates in bone marrow cells. So there's a lot of interaction with infected cells and immune cells in the bone marrow. Okay, so in order to do microscopy, you have to fluorescently label your cells that you want to see. And we wanted to see Tregs, CD8, cytotoxic CD8 cells, and virus-infected cells in these mice. And I will show you some movies, at least for two of these cell types. We have now just established to also see virus-infected cells in the living mouse. So the Treg, to find Tregs by microscopy fluorescently labeled was easy because we have the Treg mice. And I told you that all Tregs in these animals are green. So we already had green Tregs. What we had to establish was red CD8 T cells. So we had to do some mouse crossing. There's a so-called TD tomato mouse that has green cells, and green T cells, and we crossed that with T cell receptor transgenic mice that have T cells that are specific for the friend virus epitope. So when we transfer these red CD8 cells, they are all specific for the virus. And then they expand in the bone marrow and we can film them. And last but not least, we have infected cells in the bone marrow, and we can now also visualize them with a third color because we use fluorescent viruses. And when these cells become infected, they have a color. And this can also all be filmed with 2-photon imaging. And you can look for cell interactions and the motility of cells. And you can also manipulate the system by injecting antibodies or depleting regulatory T cells, for example. So this shows you the imaging of the white is the bone, blue is blood in the bone marrow, red are the friend virus specific CD8 T cells, and in green, and you can only see a few tiny green spots here, are the Tregs. They are not fully labeled, they are only labeled in their membrane. That's why they are more difficult to see. And we can zoom in here and see the interaction actually between cytotoxic CD8 cells and Tregs in the living mouse. And this is a time point 10 days or so after infection of these animals. So what we can show is that they really interact and bind to each other, the regulatory T cells and the virus-specific CD8 cells, and there's really physical contact between these cell populations. And that obviously plays a role for the functional abilities of the Tregs and also the CD8 T cells. What we can also do is we can track each cytotoxic CD8 cell, for example, and see if their movement changes when they have Tregs around or when they don't have Tregs around. That's something that we are interested in. So we can, a computer makes these cell tracks, and then we have an automatic calculation system that can show us all these cell tracks and give us speed from these cells and how long they move and so on. So here, this shows you the motility of the CD8 cells, the virus-specific CD8 cells. These are only numbers, and these are different time points after infection. So you can see the expansion of CD8 cells after infection. So at 8 days post-infection, we don't have that many CD8 cells. Then we have a clonal expansion, and we have a lot of cells at 10 days post-infection and even more at 14 days post-infection. However, when you look at their track speed, so how fast they run in the bone marrow, then you can see that the optimal time point is at 10 days post-infection, because at 8 days they have a reduced speed, or they are not so quick. And then at 14 days, they already have a reduced speed, because that's the time point when the Tregs are expanding and start to reduce their speed, actually. And high speed is correlating with a lot of CTL activity. So at 10 days post-infection, we have the highest CTL activity by these CD8 cells, which then starts to be reduced because of Tregs at 14 days post-infection. And this shows you the impact of Tregs on this CTL movement. And here are the numbers again, but let's look at the track speed that we can calculate, and especially here at 14 days post-infection. So this is here the reduced speed at 14 days post-infection, the blue line. If we then deplete regulatory T cells because we inject diphtheria toxin, this goes up again to normal values that we see, for example, at 10 days when they are very functional. So that shows that Tregs suppress the movement of CD8 T cells, and this correlates with their cytotoxic activity. And we can now answer a lot of additional questions in this model where we can follow both cell types and now virus-infected cells as well in the living mouse. So Tregs reduce the motility of virus-specific CD8 T cells in vivo. Okay, and with this, I would like to sum up the most important results that I showed you. I showed you that Tregs suppress cytotoxic CD8, and I didn't show you that, and NK cell responses. Also, NK cells are suppressed. I didn't show you the data. Transient depletion of Tregs enhances the cytotoxic effector function of CD8 cells and also NK cells, and you only need transient depletion. We inject the diphtheria toxin, and the Tregs are depleted for about 6 to 7 days and no longer. Then they come back. So Tregs suppress, I didn't show you the NK cell data, but the mechanism here is IL-2 consumption. I talked about that in the general part. So they just take away all the IL-2, and then there's nothing left for the NK cells, and they cannot function anymore. And the cytotoxic potential of CD4 cells is controlled by both cell types, by Tregs and CD8 cells. And I showed you in the last part that Tregs and CD8 cells interact in vivo. They really bind to each other. And this reduces the motility of cytotoxic CD8 cells. And this is during the acute infection, and then in the chronic infection, Treg depletion reactivates the functional exhaustion of CD8 cells, so it can be used therapeutically. And a combination of combination therapy targeting Tregs and PD-1 at the same time further improves the antiviral effect in chronic infections. So Tregs and inhibitory receptor targeted immunotherapy would probably be a potent strategy to treat chronic viral infections. And as you know, the inhibition of inhibitory receptors, that's already largely used in the clinics for cancer treatment. Okay, before I close and take questions, I have to thank a few people here, especially Gennady Zelinsky. I already mentioned him. He's from the Ukraine, and he works in my lab on these regulatory T cells for a long time now, and he will give his presentation tomorrow, especially on the inhibitory receptors. I do a lot of this in collaboration with my friend Kim Hasenkrug from the Rocky Mountain Labs in Hamilton, Montana. That's part of the NIH in the US. And I was there in the lab for 3 years. And this fantastic Treg mouse was developed by Tim Sparwasser in Hanover. And yeah, I thank you a lot and for your attention. And I stop this presentation and I take questions.
Okay, thank you very much, Professor Dietmar, for very informative lecture. And I have a question. I'm not a specialist in immunity on this stuff, but still, is it possible to completely eliminate viruses, especially in chronic virus infection and especially in herpes virus infection?
Yeah,
My husband is suffering from this disease and it appears again and again, and no treatment is helpful. Thank you.
Yeah, it's like I showed, it is very difficult. We are working on this for many years now, on retroviruses, also on hepatitis viruses, but the same is true for herpes viruses. We still don't know how to completely eliminate them. I think we have some idea how to approach this, but until this can be used in the clinic, I'm afraid it's still a couple of years ahead. But now we understand some of the mechanisms, how they actually become chronic, and now we can target them. But like I said, we are not as far as in the tumor field, unfortunately, and we need some more research to bring that really into the clinic.
Thank you very much. Maybe some students have questions or have something to say.
Maybe before we start with the student question, I just see Joachim van Drey. He's our Vice Dean for Learning and Teaching. We don't want to take too much of his time away. So maybe, Joachim, you can say a few words and then we start the discussion with the students here.
Yes, thank you, Ulf, and welcome to everyone. Actually, I have to apologize because this hacker attack still has mixed up my calendars, and I mixed up Poltava time and German time, so I was 1 hour late. So I'm really sorry, because I think this is a fantastic opportunity, and I thank Ulf and Gennady Zielinski for setting this up. I had a look at the program, and I just listened the last hour. So this is fantastic, and as a start, this technology we have now had to train and to learn during COVID is now having a big advantage, giving opportunity also to involve you in these lectures. And the program is really fantastic. I can tell you, I have given this to all my students and doctoral students, so they may somehow link in and listen to this lecture. We had this opening address last week with your rector and the representatives from your university, and if we can in this way help you to maintain your studies and even to have some really specialists from our university, it's fantastic. So I see there's quite a large group of you listening, and I'm sure that you will enjoy this whole Winter Academy, and that it will motivate you to internationally make links and even come to other countries for time. So I'm in the students team for the medical faculty, and we also try to help medical students wherever it is possible. As you might know, it is always for studying medicine the problem of language in Germany, because it's all teaching is in German and the patients are in German, and so this is actually a big problem. And currently, I'm very happy that we are working on one student from your university who has learned a lot of German, and we are just trying to pass her through the language exam and then try to somehow allow her to continue her medical studies at our university. We have connected the German universities, and some universities, as you might know, have even made courses in Ukrainian language. But to have people to continue study in Germany, it depends on the language. So that's something that we all work on, and there are language courses and so on, so that that is still something that only a very few can or are helped. And so in this way, this academy is a wonderful thing for you to have this in English, which is easier, and to have the specialists in the field. So I hope you enjoy this. And again, thank you to Ulf and Gennady Zielinski for setting that up. I think this is fantastic. And it's very good for a partner university to have such conferences and courses. So you know at least some faces from the lectures, and then maybe you have the opportunity to come and visit the labs and the institutes one day. Thank you.
Yeah, thank you very much for organizing such a fantastic Winter Academy. And I believe the students are very excited and motivated. And of course, it will be very helpful in their studies. Thank you very much.
Okay, thank you, Joachim, for holding on so long and listening to my talk. And if you have other duties, so see you again.
Thank you.
Bye-bye. Thank you. Okay, I'm open for questions now. Sorry, we had for this.
I have a question. Do we have some drugs based on T-rex depletion to treat chronic disease?
Yeah, that's a good question. So there are a few drugs that can deplete or inhibit regulatory T cell functions. There's, for example, one drug where an antibody for CD25 is coupled to a toxin. So when you inject this drug, then the target is always CD25 high cells. So you actually get a lot of Tregs with that, and the toxin then depletes these Tregs. So that's a drug that's being used in humans, and that's approved. And there are also some other chemical drugs that interfere with the function of Tregs. What we don't have so far are really good drugs that expand regulatory T cells and autoimmune diseases, for example. There are some drugs that might do that, but yeah, this is still something that we have to do more research on, because for both ways, for depleting Tregs in humans as well as for expanding Tregs in humans, we don't have the optimal drug yet. That's still under development.
Thank you for answer. And so we can possibly treat the retrovirus disease by depletion, Tregs depletion. We can treat the retrovirus disease.
Yeah, I mean, this is what we currently have. As you know, for HIV, we have antiviral drugs. So there's ART. So that's a very potent therapy that HIV-infected patients receive. But the problem is with this therapy, you cannot eliminate the virus. So you have to treat patients lifelong. As soon as you stop with the antiretroviral therapy, then the virus comes back up. And I mean, the idea would be to combine this antiretroviral therapy with a modulation of the immune system, and then achieve elimination of the virus. This is, for retroviruses, really difficult, and we don't have the optimal protocol for that yet. We also trying to do that for hepatitis B, because there we have the same problem, chronic lifelong infection. And there we have made bigger advances in the past. So there are some initial clinical studies where we can actually eliminate hepatitis B from patients. For HIV, it's much more difficult because the virus is integrated into the genome and so on. But there are studies where we try to eliminate these viruses. And for hepatitis B, it looks quite successful.
It's good. Thank you for answer. I understood.
Yeah, thank you for your question. It's a very good question. Are there more questions?
I have no question, but I thank you for your interesting lesson about T cells and presentation about Essen University. We hope that in future we could travel to the Essen University and take part of our lesson. Thank you very much.
Yeah, you're very welcome, and we would love to welcome you here in Essen. Our Vice Dean just said that there's always a difficulty with studying for people who come from abroad in Germany because the medical studies are all in German, and obviously the patient contact is all in German. However, here at the institute, so if you come to an institute for virology, for example, all our language is English. So we have a lot of foreigners here, so we do everything in English. So language is here not a problem at the institute.
Thank you.
Are there more questions? You can also then, is there another one? Igor?
No.
No.
No, there are no questions in our students. I think you had a very good start, and really it was amazing lecture. And to my mind, this lecture was too difficult to our students now. Maybe you can make next lectures little bit easy to understanding, because many phenomena, many titles of the cells or substances like OVA, for example, it should be explained for the auditory sometimes, just maybe in the first time when you used these terms, but it was really very good scientific presentations which may be done in all scientific auditories in the world. Thank you very much for this high level of the education to our student, and thank you again.
I mean, that's you're very welcome. That's really important for us. So, I mean, maybe you can have a discussion after this meeting with the students of if this was too difficult and they didn't understand a lot of this, then you should tell us. For us, it's difficult to judge what the basis is, and because I'm pretty sure that most of the lectures that are prepared for the next days are at the same level, because this is what we had for the Chinese students, for example, and they knew all these basics. So you have to tell us so that I can approach the colleagues for tomorrow and the next week, that they should maybe explain some of the basics, more broad. So please let me know as soon as possible. Now we
Yes, thank you very much for your proposition. Yeah, now I plan to explain some phenomenon from your lecture, and after the lecture, I would like to say a couple of words to our students and then explain some terms.
Okay.
Okay.
But you can write me an email. So if you have suggestions to, I mean, what we really want is that after these 2 weeks, that your students understood as much as possible, and we can still adjust for the other talks for this week and the next week. So you can write me an email, and maybe we can try to adjust a few things so that it's maybe a little easier for the next days. Because like I said, it will be, so what the people prepared so far will be the same level than what I did today.
Okay. Thank you very much. Thank you.
Okay. Yeah, if there are no further discussion points or questions, then I would like to thank you again for your attention. And I hope we can address some more questions next week during the tutorial. So you can also write your questions down or send it to us by email. That's also possible. And we pick that up next Thursday. Then and Gennady will talk tomorrow. So Gennady is already with us. You can probably already try to make things a little easier on your presentation, and maybe take out some of the very specific details.
Okay, thank you for.
Okay, so I see all of you hopefully on the last day. On the last day, I would like to say farewell words, but I'm traveling that day, and I hope our train system will get me to where I have to go on time, so that I will then be there for the end of the Winter Academy. But I really hope that I can see all of you either in Germany or the Ukraine, hopefully soon. Okay, stay safe and goodbye.